Please welcome the moderator for our next session, Alliance board member and survivor, John Woerner.
So great to see everybody here. Thanks for coming back in from break. We've got a great session lined up. Before we begin, I also want to welcome all of our virtual attendees from across the country that are joining us. We're going to be live streaming this session as well as one of the sessions later. So thank you all virtually that are out there as well. I'm really thrilled to be able to host this panel discussion. We're fortunate to have Dr. Nilo Azad from Johns Hopkins and Dr. Adham Jurdi from Moderator. Please join me in welcoming them to the stage. So what we're hoping to cover today is really, what is the state of the union? What are the options that are out there beyond standard of care? What's in trial? What's coming next? And if we're giving you a fire hose, you can blame me. I said, we've got to get all this out there for folks. So we're going to cover about 4 different topics, maybe about 8 to 10 minutes each, and then have a section for Q&A at the end. So we'll just jump right in, and we will get started with immunotherapy as our first topic. And Dr. Azad, you want to kick us off? I mean, it's such a broad topic. We had great success with MSI. Now they're starting to move immunotherapy earlier and earlier in trials for MSS, the NEST trial that all of you have heard about. And Dr. Azad is kind of at the forefront of a lot of that. So please let us know what's happening.
Well, thank you. First of all, I want to say hello to everybody. This is such a great conference for scientists like me to be able to come to and really engage with the advocacy and the patient community. So I really appreciate the invitation. Over the last decade, immunotherapy has really revolutionized cancer care for so many different kinds of cancer. But in colon cancer, I think we've really struggled with the majority of patients that have colon cancer. So we know about 3 to 5% of patients have microsatellite unstable cancer, and immunotherapy has been groundbreaking there. But in the 95% of patients that are metastatic that have microsatellite stable cancer, we've really struggled. Maybe we could move on to the next slide. And so what we've really been working on is how do we define immunotherapy? A lot of times it's been kind of synonymous with an immune checkpoint inhibitor, PD-1. You've heard Keytruda or Opdivo. But actually immunotherapy is many different things because the immune system is actually quite complicated. And we have many different immune cells that we need to engage, and we can engage them in different ways. And so this slide just shows the response rates of some of these different kinds of immunotherapies. And as you can see, they do best in microsatellite stable patients right now, state of the art today, when combined with chemotherapy. But we really want to get to a point where we're going to be able to use immunotherapy without chemotherapy. Maybe we can move on to the next slide. And so this just is a little bit of a foundation in terms of our conversation around immunotherapy. So we have multiple strategies that we can use to engage the immune system. The immune checkpoint inhibitors that we've talked about, those are really meant to supercharge the immune cells. So the immune cells are already there, and they're meant to turn on, activate T cells or other immune cells. And so they're checkpoints on the surface of these immune cells, and by targeting them, we can activate the immune cells. So that's one group of drugs, but we have so many other kinds of drugs that are coming into the clinic. Another important problem in this cancer situation is how does the immune system even recognize that the cancer cell should be targeted? And so that's what in this panel B, it's about how do we figure out, we call that an antigen, the thing that is actually targeted by the immune system. So how can we, cancers are living in plain sight within our systems. How do we get the immune system to recognize it? So this is where we can use cancer vaccines, or we can use other drugs that help activate immune cells around these different antigens. And then this third panel is the idea of, can we find drugs that on one side bind to the immune cell and activate them, and on the other side bind to the cancer cell? So they bring them together, because you've got to get the immune cells to the right place. So we call those bispecific antibodies that are T-cell engagers. And then cellular therapies is a really exciting area where we give immune cells back to patients after manipulating them so that they actually are targeting different cancers. So maybe our next slide. And you just referred to the NEST trial. This is in the microsatellite unstable. So in that 3 to 5% of patients, it goes up to 10% when you go into earlier stages. This is the data about immunotherapy with PD-1 inhibitors and immune checkpoint inhibitors, which is just amazing. If you look at this, what you're seeing is each one of these lines, as you I'm sure have seen in some of the other things that have been presented, represents a patient's tumors, and those lines going down to the bottom of the graph are showing that they have a complete response. The cancers just disappear. So if a patient has microsatellite unstable cancer and you give them immunotherapy before surgery, these patients don't even need to go to surgery. And this has been an amazing development for our MSI high patients. And then I think our final slide, no, that was our final slide.
There's also been, I guess, some immunotherapy tried before surgery, right, in colorectal. I think Dr. Kazi and others. Any thoughts on that and what's next there?
Yeah, so we had thought that it was a binary situation. If you have microsatellite stable cancer, immunotherapy with immune checkpoints doesn't work. And then a small study was just reported where these same immune checkpoints were given to patients with microsatellite stable cancer, so the majority of colon cancer patients, but it was given to people in lower stage disease, so stage 2 or stage 3 disease. And in those patients, the majority of them had beautiful responses. And then they went to surgery, so they were able to take out the tumor and see that many of those patients, after just a couple of doses, the tumor disappeared. So it's a small study. It was just published in a big journal, so it was only a couple dozen patients, but that's going to now move forward in a larger trial to see if this is actually accurate, when you start going to larger trials with more patients. So this will be coming soon to trial locations across the country.
Next, we'll pivot to vaccines. Vaccines are also kind of part of that immunotherapy broader world, and they hold such great promise, I believe, and many of us believe, in helping treat colorectal cancer. They've been in research for, I think, decades. I myself, back in 21, stage 4, had just had progression on FOLFIRINOX after 2 VAT surgeries and went to search for a trial and ended up doing a vaccine trial at NIH. Almost did one with Dr. Azad, but got on the NIH one as I was a better match at that time, and served me well. But the science has come so far even in the 3 years. So Dr. Azad's led multiple research efforts in vaccines. So tell us where we are and where you see it headed.
Yeah, so we're in a brave new world when it comes to cancer vaccines, and so it's really been exciting to see how technology has caught up with what we've always wanted to do with cancer vaccines. Maybe we can move on to our next slide. So there's some things to really consider when you think, like, why do we even vaccinate? Like, what is a cancer vaccine? Why do we do this? So the idea is, like we just talked about, cancer is hiding in plain sight. It's sitting there, and the immune system is not recognizing it. So what we need, when you give a cancer vaccine, the idea is that you're amping up the immune system to be able to recognize some component of those cancer cells so that they will go there and kill those cancer cells. But there are a lot of features that we have to figure out in order for a cancer vaccine to work. So number one, what pieces of the cancer cell are actually the most likely for the immune system to recognize. So that's called antigen recognition. And so trying to figure out, that's a very large problem. And there's large companies as well as academic investigators that their entire apparatus is focused on how do you figure out which parts of the cancer cell are actually stimulating the immune system or immunogenic. And then when you give a vaccine, you have to give it with something to help increase the immune response. So that's the adjuvant component. And you guys everyone's heard about mRNA vaccines with COVID, but there are a lot of different kinds of vaccines. So what vaccines are the best? What are most likely to create an immune response? So that's a choice that you have to make. And then not only do you have to get the immune cell to recognize the cancer cell, but then it has to be turned on. So even if you get the immune cell there, if it's not turned on, then it's not going to do anything. So we, in our group, for example, administer vaccines with immune checkpoints to turn those those immune cells on. And then you also have to figure out what the right time to treat somebody is. So is it better to treat someone when they have very little disease, like in the MRD positive setting when you've got positive ctDNA, but you can't see tumors, or earlier in their therapy, or is it okay to try it in patients who have already had substantial metastatic disease? Next slide, please. And then the big thing that I really wanted to share is the idea of personalized vaccines versus shared antigen vaccines. So personal vaccines are when a patient comes in, you take their tumor, you sequence it, and you create a bespoke vaccine for that one person. So that vaccine matches that patient, and nobody else is going to get that vaccine, and it's made specifically for them. Now, you can imagine that on one hand, that could be particularly powerful because you're really harnessing that patient's tumor. But on the other hand, like when you think about scaling up to the tens of thousands of people that have colon cancer, it could be a very difficult thing resource-wise to scale up to make available to everybody. So, and then on the other hand, there are things where, are there certain antigens, certain parts of the cancer that are shared among all people with colon cancer or most people with colon cancer? And could we create a vaccine to that so it's just off the shelf? It's like any other drug that you could get at the pharmacy. And that clearly has significant advantages in terms of, hey, you come in, if you've got this protein, you get the vaccine. But on the other hand, it's not as perfectly personalized. And so what we have been focusing on, and I think what we're going to share in a couple of slides right now, are the idea that there are shared antigens that can allow us to vaccinate many patients, but that we are still able to create an immune response against. In particular, KRAS mutations have been that space. So about 40% of colon cancer patients have a mutation in the KRAS gene. And we and other groups, which we're going to share some data right now, have created vaccines that target KRAS. So you could give this to any patient that has a KRAS mutation. So this is a vaccine that is by a company named Elicio. And I think it's really exciting data where they basically created a vaccine against a couple of different KRAS mutations. So this covered about 20% of patients with colon cancer, this one off-the-shelf vaccine. And they did a clinical trial where they took patients who went to surgery and they got chemotherapy after surgery. And then they said, after that, we're going to vaccinate patients if they have some high-risk feature. So they did a blood test and their CEA is still elevated, or they did a blood test and they had positive ctDNA, like a Natera positive test. So then we're going to vaccinate those patients because we know those people are at high risk for their cancer coming back because their markers are still elevated. And what they showed, and that's what you can see here in this waterfall plot, is that many of those patients were able to clear that high-risk feature, like their CEA went down or their ctDNA went away or decreased. And so that was really proof that we can create a response with this vaccine that can help patients. Next slide, please. And this is a study that we've done at Johns Hopkins, and this is a trial that I've led and I've been working with Liz Jaffe and Neha Zaidi, who are two other investigators at Hopkins who are really focused as laboratory investigators in this space. So we were really interested. Our mission at Hopkins has really been predominantly focused on patients with metastatic disease, and so we were interested in seeing whether we could use a KRAS vaccine for patients that had advanced disease. So we took patients who had already gotten chemotherapy, and we treated them with our KRAS vaccine that covered almost all the KRAS mutations that patients with colon cancer can have. And we also gave them immune checkpoint inhibition with nivolumab and ipilimumab, so a PD-1 and CTLA-4 inhibitor. And I'm going to show you one slide of unpublished data, but just because we're so excited and we thought this is the right audience to share it with. So if you could just show me the next slide. So we're seeing responses in our patients. So these are in people who you would expect a 0% response rate. And you know, if any of you have been hearing about botensilimab and balstilimab, which is, I think, a really exciting immune checkpoint combination in colon cancer, they're only seeing responses in patients that have lung metastasis or that don't have liver metastasis. Majority of patients with colon cancer do have liver metastasis, and if you look at this figure, all of the green bars are patients with liver metastasis. And so we're very excited about the fact that we are seeing responses in patients that have heavily pretreated disease. So these are people who would really have one or two options left in terms of treatment, and instead with this immune approach have really had substantial reduction in their tumors. And one more slide. And so we are really moving this forward for patients in the first-line metastatic setting now. So we're now doing this trial with botensilimab and balstilimab. So we're trying to move into the more novel and next-generation immune checkpoints, but we're also combining this with vaccines, and we're giving this to patients right after they finish their first FOLFOX or FOLFIRI in the first line, really hoping that if we move this earlier that we might even be able to think about that cure that is sometimes possible with immunotherapy. So Elicio continues their work as well. They are really deployed more in the adjuvant setting, so in stage 2, stage 3 disease, and so we're right now more focused on the metastatic setting. And so hopefully with many people working in this space, we can really work for all different kinds of our colon cancer patients.
And is this the same mRNA vaccine as the earlier trial?
This is a peptide vaccine, but yes, it's the same peptide vaccine as the previous trial.
And is there a theory? Is there a theory why is it that this particular type of mRNA vaccine might work better in metastatic disease versus MRD positive disease, where some of the other ones are trying?
I don't think that we can say, and it probably doesn't work better in metastatic disease, but we just still, as I said, our mission really is in the metastatic setting, and so we just, that's the area that we really wanted to focus, and it very well many things seem to work better in earlier stage disease, and I think the use of vaccines in those settings are totally appropriate and very exciting. Just, each one of us are kind of looking at this puzzle in a different way.
And Dr. Jurdi, a number of these vaccine trials are looking at patients who are NED, maybe stage 3, maybe stage 4, but ctDNA positive.
Right.
Can you talk about how you think about getting those people to flip to negative and what that might mean?
Right. So it's a really promising approach, and the idea, Nilo touched on it, but the idea is, excuse me, instead of trying to stimulate your immune system with the vaccine to attack chunks of tumor, you're really treating it when there's nothing on scans and it's only MRD, so molecular residual disease. So it's kind of a lighter lift for the immune system, so to speak. Now, there are some nuances to it as well. Vaccinating while the tumor is still in there can produce a more robust immune response. But for the ctDNA portion, the AllyCon trial we just saw, it did use ctDNA as an enrollment criteria. So patients were only receiving the vaccine if they had completed all treatments and nothing come back on scan, but their ctDNA test was positive. And then 6-month clearance of ctDNA. So lack of detection of molecular residual disease after 6 months of treatment was the primary endpoint. I think that has a lot of promise for all future trials, and I'll touch on that a little bit as to where the regulatory landscape is heading for clinical trials for ctDNA.
Yeah, let's jump to ctDNA as our next topic. Signatera testing, other ctDNA testing, I think is critical. I advocate that patients use it at various different stages. Dr. Jurdi with Natera is kind of right at the center of the innovation in this space, and it's fascinating research we've seen over the last 5-10 years coming out using this as a tool. When I was first stage 3 in 2018, I didn't even know about ctDNA testing and learned about it in 2020. It's a great asset if you're not plugged in and thinking about using that in monitoring your cancer. But Dr. Jurdi, maybe bring us up to speed on where we are and where we're headed.
Yeah, absolutely. So I'm very fortunate to be really at the epicenter of the ctDNA, I want to call it revolution, but in a way it is, because it's going to be a whole complete paradigm shift in how we manage cancer. First of all, I do want to say thank you, John and CCA and AllyCon, for the invite. As a clinician who's not in clinic anymore for the past 3 years, patients and patient advocacy are near and dear to my heart, so it's been great meeting some of the attendees and hearing their stories. So what we're showing here is really kind of a simplified timeline of how did we get here from ctDNA. So cell-free DNA, or just circulating nucleic acids, was described as early as 1948, not specifically for cancer, but just like any cancer, any cell in the body shedding nucleic acids. It wasn't until 1994 where we started showing kind of accelerated signs that we can actually do this and test for cell-free DNA. And it wasn't until 2006 when ctDNA, or circulating tumor DNA, was first described. But then you'll see, like, from 2012 onwards is when things really started to accelerate. And there's a couple of reasons for that. The main reason is really technological advances. So sequencing became much cheaper. It became more practical and feasible to use it as a method for detecting ctDNA. One of the first studies in colon cancer came out in 2016. Dr. Jeannie T out of Australia. It was really a seminal article in that space. In 2020, for Signatera, our test, that was when we published our first validation data set. And that led the following year in 2021 to FDA breakthrough device designations, as well as coverage by Medicare for stage 2 through 4 CRC. 2022, Dr. T again presented the dynamic study, which was published in New England Journal, which was the first randomized study that they used for one arm, just standard of care management, and the other was ctDNA guided management. So patients after surgery, specifically stages 2 and 3, if they went on the ctDNA arm and they were ctDNA positive, they received adjuvant. ctDNA negative, they went on observation. And it was a positive study. It was more of a non-inferiority study, so it did meet its endpoint. It did have some criticisms about the design, but I think it was very eye-opening. And fast forward, last year, Signatera, we published in Nature Medicine showing for the first time the predictive value of ctDNA for adjuvant chemotherapy. And just a few weeks ago, we saw that the FDA issued the final guidance for industry that ctDNA can and probably should be used for designing future clinical trials. So whether you're enriching your clinical trial with just ctDNA positive, so instead of treating all comers with a new drug or vaccine, whatever it may be, you're really focusing on the small portion who are ctDNA positive, because those are the ones with residual disease where you can actually change their outcomes, versus the negative patients, which we call it diluting the signal. You're treating a lot of patients with these new agents who may not need it. And in fact, if you can't detect the cancer, are you actually, what is there to respond in a way? So it's really encouraging to see the FDA on the forefront of this, and the idea is to have ctDNA clearance after therapy as an endpoint for trials. So instead of waiting for 2-3 year disease-free survival, 5 year overall survival, we're able to get an early signal with ctDNA. So in a way, we're running smaller, faster, leaner, cheaper trials with a very enhanced signal that is being amplified by ctDNA.
As you think about where we're headed, do you want to talk about your, am I not advancing there?
Yeah, actually, if we can go back, John, real quick. Just fast forward to today, and we just celebrated, actually, last month, over 100 peer-reviewed publications on Signatera across different tumor types. This is over 6,500 patients. And I just want to highlight that this all happened in the past 4 years. If you look at this timeline, there's obviously the need and thirst from us in clinic and for the patients to have a better biomarker to guide our management. And that's why you're seeing that more than 40% of US oncologists have ordered Signatera to date on over 250 patients. And as a byproduct of all these publications, we have Medicare coverage beyond CRC to breast, bladder, ovarian cancer, and really any solid tumor on immunotherapy. So it's really good at picking up response to immunotherapy early on. Yeah, so just really briefly, when we think about ctDNA, there are two approaches for the commercially available tests. One is the tumor-informed assay, and that's what Signatera is, and beyond tumor-informed, it's also personalized. So the way the test works is, as the name suggests, it is tumor-informed. So we start with tissue, be it from a biopsy or a surgical specimen. We do whole exome sequencing of the tumor tissue, which kind of gives us the blueprint of the cancer. From there, the algorithm picks up to 16 mutations to track, and those 16 mutations become that tumor's barcode or signature, so to speak, hence the name Signatera. So you only need the tissue the one time. From there, it's a 16-plex PCR assay that you can just use it as many times as you want. Not as many times as you want, but meaning despite potential multiple recurrences, the test should still pick up molecular residual disease if it's coming back.
To that point, when people are monitoring their cells, like I am, I do it every 3 months. Is that
Right.
what most patients would do if they're NED but monitoring for a potential recurrence?
Correct. Depends on somewhat on the stage, but more so on where you are in the cancer journey. So in the first 2 years, definitely every 3 months. The first 6 months, actually, after surgery, we recommend 4 tests. So I like to think about it as 1, 1, 2, 2. So 1 month after surgery, 1 month after that, 2 months after that, so month 4, then month 6, 2 months after that. So
That's helpful.
You get a little bit more of an intense monitoring because we know if it's going to come back, that's the first 6 months is the highest risk of the cancer coming back. And we want to pick it up as early as possible. Once you go beyond 2 years, we think 6 months, every 6 months is reasonable. We still need more data there, but for the first 2 years, definitely every 3 months.
Thank you.
So switching over to tumor-naive MRD assays. So here you don't need tissue. It's just a blood test, and the way it works is there are 2 approaches here. One is what is called the hotspot panel. So just a fixed panel that tracks common mutations, for example, KRAS, APC, et cetera, in colon cancer. And the second approach is to use what is called methylation or epigenomic signature. So those are changes that we see on the cell surface, well, on the DNA, and we can look at that methylation pattern and say, this is coming from colon, this is coming from breast cancer, et cetera. The tumor-naive assays have the advantage of being faster for the first time point because you don't need to custom make a test for each patient. It's off the shelf, but that's the pro. The con is in terms of sensitivity and specificity and the performance of the ctDNA assays. So far, all the data has shown that tumor-informed is a superior test and is more likely to give us more accurate results.
One setting where I see patients using that is they may be newly diagnosed stage 4. I urge them to say, can you get a blood draw done with the ctDNA because they can come back quickly, often with what your mutations are. And now in the world, there are some first-line metastatic CRC trials that can inform them whether they would fit, right?
Yeah, so just one note here to highlight is it is also a liquid biopsy, and it is ctDNA, but it's not an MRD test. So when you think about liquid biopsy, broadly speaking, there are two applications. One is MRD, or molecular residual disease, which we just talked about, and the other is to look for targetable mutations and pick that up from the blood and to be able to use some of the more novel agents.
Great.
So it's a different test than Signatera. It doesn't provide that, but there will be that.
And I just wanted to say, like, Natera is a very important company in this space, but there are multiple other companies that are working in the liquid biopsy, liquid ctDNA space, and they each may have different benefits. So we're going to talk about KRAS in a moment, but, for first-line clinical trials where we really need the results very quickly, some of these non-tumor informed or liquid biopsy tests can be very helpful to get results back within a week to be able, if you've got a new diagnosis of cancer, you may not be able to or want to wait 1 month for your personalized assay. But for surveillance, the personalized assays definitely, in today's technology, function better than the non-tumor informed. So there is a role for them, and working with your doctors to figure out which test for your particular situation is important.
You can talk briefly about in stage 2 and 3.
Right. So
Escalate.
Yeah, as we just said, we're over 100 publications, so a lot of data, but I tried to distill it down to a couple of take-home points, especially from this paper that just came out in Nature Medicine a couple of months ago. So this is coming from the Galaxy trial, which enrolled over 6,000 patients. It's the largest ctDNA platform in the world to date for CRC. And the most recent paper reported on 2,200 patients. So I'm focusing on high-risk stage 2 and 3 because that's the population where, as clinicians, we need to decide who needs adjuvant chemotherapy, who doesn't. Because we know we overtreat patients, right? Stage 2 colon cancer, surgery cures 8 out of 10 patients. 8 out of 10 patients. And the benefit of chemotherapy is at most 5%, yet we treat about 35 to 40% of stage 2 patients with adjuvant chemotherapy. And then for stage 3, right now we say anyone who's stage 3 should receive adjuvant chemotherapy. But in reality, not all stage 3 are created equal. You have people all along the spectrum. Low-risk stage 3 can be as good of a prognosis as stage 2, or maybe even better than some of the stage 2. So what we're seeing here is when we look at just stage 2 and 3 and look at ctDNA just 1 month after surgery from a single time point, and we divided the patients or stratified them by who received adjuvant chemotherapy and who went on observation. So what we're seeing on this slide is patients who were ctDNA negative, and the lines you're seeing are completely overlapped. So what that means is patients who received adjuvant chemotherapy or patients who went on observation, they did the same. Chemo did not add a survival benefit. You'll see the numbers that it's not statistically significant. And those patients have excellent prognosis, over 93%, 24-month DFS in either case for stage 2, and a similar pattern for stage 3. So one caveat here is this was not a randomized study, but 2,000 patients is 2,000 patients. The randomized portion of that trial is ongoing. We probably won't have the results until early 2026. But beyond Galaxy, we've shown in multiple trials, as well as independently published research, the reproducibility of this. So yes, it's non-randomized, but when you have over 10 or 15 reports all showing the same things, we believe that in the randomized trial, the results will hold up. On the flip side, if your patients are MRD positive, that just tells you there's disease there. Right? So those are definitely the patients you want to treat. Now, whether the cancer will respond to the treatment, to FOLFOX or CAPOX, that's a different question. But what we're seeing here is patients who were MRD positive, if treated with chemotherapy, that's the top line that you see. So at 24 months, more than half of the patients who received adjuvant chemotherapy in stage 2 were alive and disease-free. versus just 10% with observation. And with stage 3, it's even worse. So 33% if they received chemotherapy were disease-free at 2 years, versus really no one reached the 2-year mark in the observation arm. So if your patient is MRD positive, and here this studied high-risk stage 2, but even low-risk stage 2, ctDNA is the most prognostic tool we have in our hands today after surgery. So even if a traditional low-risk stage 2 is MRD positive, that puts him way higher risk than the rest of the stage 2s. And to that end, we've shown a lot of data, but we continue to generate evidence. I do encourage everyone who is eligible or knows someone who's eligible to enroll on the Circulate North America trial, which is a study really being done by the NRG group in collaboration with Natera. And here, patients after surgery, if they're ctDNA negative with Signatera, they get randomized to standard of care adjuvant versus observation. And if they're MRD positive, they get randomized to FOLFOX or CAPOX versus FOLFIRINOX, which is the escalation arm. And even patients on the negative arm, if they turn positive during surveillance, they can get re-randomized to the positive arm. So the study's open across the US and Canada. It's enrolling well. We expect, well, I don't want to say when we expect, but we hope to complete enrollment sometime in 2025. But of course, it'll be a little bit later to get the results.
Then what do you do if you're ctDNA positive after that adjuvant time
Yeah,
frame?
so great question. So I'll have 2 answers for that. What do we know today, and where do we want to go? So what do we want to know today is what I'm showing. So on the left-hand side, you'll see results from MD Anderson's INTERCEPT program, which reported on over 1,000. I believe they have over 2,000 now. And what they were looking at is patients who, during surveillance, they turn Signatera positive. What happens then? Well, when they did the first set of scans, they identified recurrences in, I can't read the exact numbers here, but it's somewhere around 40%. There were 19 patients who had suspicious findings, and the rest were completely negative. The negative ones, by the way, they're using that as a pipeline to enrich for MRD trials, which is also something we need to talk about. But if the CT scan is negative, the next step would be to do an MRI or a PET scan, and that out of the 19 suspicious findings on CT scan, 18 were confirmed as recurrences. So long-winded way of saying, right now, the main benefit is to intensify surveillance in hopes of picking up oligometastatic recurrence, where it's only a couple of spots, which again, in the hands of the right multidisciplinary team, we can decide whether we want to cut it out, radiation, chemotherapy first, et cetera. Also, we reported on Bespoke CRC earlier this year, our first interim analysis, and out of 100 patients who recurred, 40% received oligomet-directed therapy. That's a pretty high percentage. The data is kind of all over the place, but 20% is usually what we see patients who receive oligomet-directed resection. Also depends on where you live, et cetera. So we are able to detect oligomets early on and give patients a second shot at cure with oligomet-directed therapy. What to do today if you don't find anything on scans? Enroll in MRD trials. That is how we're going to generate the evidence that if we pull the second line of therapy early on, not wait for the cancer to come back and establish on scans, if we give that patient the drugs when it's still, again, at a low level, just on a molecular level detected, can that improve outcomes? So definitely, I encourage you to enroll on a lot of trials. There's, CCA's website has it, guidemrd.com also is a great resource.
If I could add to that, just as a patient, right, I'm one that if I see anything, I want to do something. And I talk to a lot of patients who might flip positive. They're NED, but they flip positive, and the doctor says, well, there's nothing we can do until something shows up in the scan. In one sense, they're absolutely right. There's nothing approved, there's nothing proven that treating MRD positive disease gets rid of your cancer. And but as a patient, I'm in the camp of, if that happened to me, I want to try, and I want to help advance the science. And these MRD trials help us prove it out. Does it work or not? And so, as a community, we're only going to figure that question out if we participate when it makes sense for us.
Absolutely. I mean, the proof is in the pudding. There's a lot of irons in the fire right now. Some will start reading as early as next year, but that's how we're going to move the ball forward.
Great. Let's hit our last topic, which is targeted treatment, specifically on KRAS. Those of you who have been in the colorectal community for a number of years know that for so long KRAS was untargetable. Then we had the great breakthrough with KRAS G12C, which was great. Then they figured out they needed to combine that with cetuximab to get more deeper and persistent response. But as we were preparing for this talk, I was talking to Dr. Azad, I'm like, we've got to tell them all the trials that are out there they could think about. And the great news is we couldn't fit them on a slide. It is so amazing how much is coming to clinical trials in KRAS across mutations. So Dr. Azad will kind of give us her view on where we are now.
This is an extraordinary time in cancer care and in new drugs. And I don't know that there's any class of drugs that has been more exciting in the last couple of years than KRAS inhibitors. And so if we can move on to this slide. Yeah, so basically, when we think about our DNA, our cellular DNA, cancer mutates the DNA, right? And so KRAS mutations are the most commonly mutated oncogene, so a gene that causes cancer, in all solid tumors. And yet, for 40 years, it was identified in the 80s, for 40 years we have been completely unable to target this gene. And we've tried in many different ways. We've tried to hit it, we've tried to hit downstream of it, meaning the signaling cascade that it results in. All of those have been unsuccessful. And then, a few years ago, the next slide, what ended up happening was that a brilliant laboratory was able to identify one of the reasons it was so hard to target KRAS is that the mutated protein is like a ball. So it's really hard to target a ball because it's so smooth. But with G12C mutations, a particular mutation, it's only present in a couple % of colon cancer, it made a divot in the ball. And so the Showcat lab was able to create a molecule that fit right into that divot and turned that gene off and that protein off. And so it was extraordinarily exciting. It turns out that gene, that G12C, is more common in lung cancer. And so it was very effective in lung cancer. If we want to look at our next slide, we looked at it in colon cancer as well. And it also was quite effective in colon cancers. This is a drug called adagrasib. There's 2 drugs that have been FDA approved in this space, but so adagrasib had benefit in terms of tumors shrinking. And then when cetuximab was added to it, John, like as you were saying, that actually improved even more. But it's 2 to 3% of colon cancer patients that have this particular KRAS mutation. So it's exciting, but we want things that are going to work for many, many more patients. And so in the last few years, that's finally happened. So if you look at our next slide, we have many drugs that are being developed in the KRAS space, but I wanted to highlight this particular drug for a couple of reasons. First, it's the most advanced, meaning it's been in the clinic the longest and the most patients have been treated with it, a few hundred patients now. And secondly, it hits all KRAS mutations. So it's a multi-RAS inhibitor, and it works in a different way than trying to fit into the divot. It actually works because there's a couple of proteins that work together, and it is involved in inhibiting that interaction. And so this drug, which right now doesn't have a name, so it's RMC-6236, but it will have a name soon, has been looked at in people that have colon cancer. This is a small group of patients, but if you see every single one of those bars is heading south, which is great. That means tumors are shrinking or being under control. The disease control rate was over 90% Again, remember, this is heavily pretreated metastatic colon cancer. So we're talking about patients that don't have very many options, and when they got this drug and they had actually already been on a G12C inhibitor, they had substantial benefit. Now, I mentioned that colon cancer, 40% of colon cancer has a KRAS mutation. Turns out pancreas cancer is 90%. And so the majority of the development and the data that exists right now is in pancreatic cancer, but similarly, they're seeing these results in pancreatic cancer. So they're seeing 90% of people with disease control with pancreatic cancer, and now the companies are opening up a lot of arms of colon cancer as well. So we're going to see more data emerging in colorectal cancer, and we fully expect that it's going to match the exciting data that's been in pancreatic cancer as well. Let me look at our next slide. And so this is a figure from a paper from earlier this year, and I put this incomplete across it because it's already obsolete. But there are many different ways that KRAS inhibitors are, or KRAS is being inhibited now. So these drugs are called direct KRAS inhibitors, the ones I just talked about. At the beginning of our hour, we were talking about KRAS vaccines. That's just a different way to target KRAS. And then there's a lot of other drugs that are working on how to degrade this KRAS, which is a protein that we want to get rid of in these cancer cells. And so really we're just, our next slide, you can see we're really in a position right now where for patients that have these mutations, we have just an extraordinarily high number of options. So if you or anyone that you know has a mutation in KRAS, really people should be looking for clinical trials in this space. These are active, exciting drugs, and the biggest quandary is how to get more patients access to them.
Yeah, and this, there's so many trials out there now in novel combinations. They're taking things in the red box, the direct targeting, and combining with upstream, downstream, chemo, other immunotherapy options to figure out what works best. I have a kind of a simplistic belief that, you know, if we can hit the cancer multiple ways, it increases the odds that perhaps you'll get a deeper response. And
Absolutely.
So I say explore these if you're in this situation.
Absolutely. There's some really beautiful work that has come out of the laboratory where patients that are given, mice, okay, so mouse patients, but mouse patients, when they're given KRAS inhibitors, that their tumor microenvironment, so the area around the tumor, completely remodels in a good way, basically in a way that allows us to think that immunotherapy would work better. So there is now really nice data that suggests that immunotherapy combinations with KRAS inhibitors are actually rational. They make sense because you can remodel with the KRAS inhibitor and then come in and add the immune system in, and that's like the 1-2 punch that you were talking about. So we actually, you know, we run a lot of KRAS inhibitor trials at Hopkins, and one of the things we've done is we've added an arm to that vaccine trial that I just showed you guys, but that arm is for patients who've had a KRAS inhibitor. So they've previously had a KRAS inhibitor. Our hope is that they've been remodeled around the tumor, and then we're going to come in with immunotherapy, and we hope it'll work even better. So those kinds of strategies, I think, are particularly exciting.
So we've thrown a lot out there. Hopefully that was a lot of great ideas and thoughts that might help you in your treatment journey. Let's open it up to Q&A for additional questions for Dr. Azad and Dr. Jurdi. I'll do my best to see, but is right up front here.
Good morning. My name is Jenna, and my question is about Signatera. So I was on a different DNA tester in 2022, and I was getting it every 2 months, and my doctor said that it wasn't approved for metastatic colon cancer patients with multiple recurrences. But, you know, my doctors try to just do everything they can. So we were on that, but then the cancer came back, even though all of my tests came back negative. So at that point, we did switch to Signatera. I take that test now. So far, so good. So I'm just wondering, is that one approved for metastatic colon cancer patients with multiple recurrences, and how effective do you think it is for those types of patients versus stage 2 or 3?
Okay, great question. So it is approved for oligometastatic stage 4, technically not patients with multiple recurrences, but we already have shown data that doesn't matter multiple relapses, it is able to pick it up. In fact, some of our data that was presented earlier this year, an increase of 10 to 15%. Now, this is not etched in stone, but an increase of ctDNA of about 10 to 15% is a strong signal that the disease is progressing, even though the scans might take a few weeks to catch up to that, or sometimes longer. And also at ASCO GI next month, we are going to present Signatera's performance in metastatic CRC through different lines of therapy, so that'll answer more of your questions. The second part of it is, even though it's not Medicare approved, patients and clinicians are using it for that reason all the time, for treatment response monitoring. And as a company, the policy right now is we don't want any patients to get stuck with bills. So you shouldn't get balance bill, even though if it's a non-covered indication, we just wanted to put it in the hands of patients and doctors and generate the evidence in a real-world manner as well.
Additional questions? Here we go.
I just got my negative Signatera result this week, so thank you very much. As a stage 3C survivor, the information is power. And one of the things that I want to talk to other patients about now as a survivor when I'm advocating and advising is how important getting all of that information is. And I think, Dr. Woerner, you hit it spot on, right? That's the whole reason why we want to include this in our surveillance. As other ctDNA options come out, especially the difference between the personalized ones and the non-specific ones, with where things are now, there's already this challenge of trying to convince oncologists who aren't in that 40% that have ordered it. I heard that same thing. Well, it's not going to change your clinical treatment, so why do it? Well, I'll tell you, I don't care if it changes my clinical treatment. It's information I want, and that gives me peace of mind, and that peace of mind is clinical treatment. Now that there's other options, how do I talk to other patients about this and educate them and let them do the same kind of homework and make informed decisions so that they can go to those oncologists who might not be out front and speak with some degree of certainty about what they want and what they should be asking for?
I can jump in. I'm not a doctor, but I'm a fellow patient, and when I'm talking to other patients, what I say is, tell your doctor that you want this test because you might think about an MRD trial if it's positive, and you don't know unless you have the test. And usually the doctor says, okay, I'll order it then. I don't know if you have any better advice.
I think that's probably the best hack,
Yeah.
in a way. Of course, we're happy. I mean, we have our patient advocates. We're happy who can connect our medical affairs team with a specific oncologist and provide some additional education. We're happy to do that. But I think just asking for it and saying, I'm not saying I want you to act on it, but I want that data. But Nilo, I'd like to get your thoughts as well.
Yeah, I mean, I have the benefit of working at a major academic center where, so my guess is that the reason that anybody gets pushback is because of some of the burdens of if it's denied, right? And companies like Natera and the other companies as well often will step in and bring that copay down or even eliminate it altogether. But there's a lot of rigmarole that a private oncologist office has to go through when anything is denied, medications, and these testing. And we do see denials for things that don't have a proven change in your care, as much as it does provide knowledge, that is power. And so I think that's my guess, is that if people are giving or reticent, it's because people are busy and they really want to focus all their time on what can provide care to their patients and these extra. And so I think that's part of advocacy too. And I think it's important for us to be advocating around some of these insurance hurdles that really take away from the patient experience and the kind of knowledge that you can get from these assays. I think we've learned a lot. I'll tell you, I was pretty skeptical when ctDNA came about. Not skeptical about ordering it. I actually ordered it all the time because I thought we needed to learn about it academically. And I think that it's about what you're trying to get from that, right? So if you're trying to get information about ctDNA changes so that you diagnose something early, like the bespoke study showing that we can actually move people more towards cure, now you've got something that you can say there's something tangible that's going to come from this. So I think in the next couple of years, you're going to start seeing a lot of changes in practice, and we're just at the beginning of it right now.
Question in the center in the back.
Hi. Thank you so much for all you're doing to help cure cancer. I'm here with my sister who's been fighting colon cancer mucinous type for 5 years. And we just had a question around, with the mucinous type, the Signatera has not indicated when she's had a relapse. So just wondering if there's any information, research being done on any sort of test that can look at that with when it doesn't, I know mucinous doesn't shed as much DNA. And then the second question is, her genetic mutation is KRAS wild type and NRAS amplification. And just wondering if those targeted treatments work with that. Thank you.
Go first.
So I'll take the first one. So yes, mucinous adenocarcinoma, as the name suggests, has a lot of mucin. So the problem is the cellularity there is low in the biopsy. So the input that you need to generate the test is low. So other ctDNA approaches are probably going to face the same issue. We've seen this specifically with appendiceal cancer, which is more likely to be high mucin, where the sensitivity is not great, and that's just a biological limitation. So I haven't seen any data from any other approaches that has shown improved detection in mucinous cancers.
Although I will say there's hope, because I read all the time, there's literally dozens of additional ctDNA testing companies working on developing innovative tests, and I know you're innovating as well. So I'm hopeful there'll continue to be a proliferation, and then the scientific community can help figure out how to optimize to the best result. And then I think there was a question
About
also.
NRAS.
NRAS.
Yeah, so, it was interesting. So there's a concurrent scientific meeting happening right now, and we were just talking about NRAS there because the multi-RAS inhibitor that I mentioned does have some NRAS activity as well. They have not opened a cohort for NRAS mutation positive or amplification positive patients yet. We are advocating for that. I think a lot of times our rare biomarker subsets get forgotten, and that's really unacceptable. And so we've been advocating for that. One of the good things about a million companies in this space is that they have to compete with things with Revolution Medicine's drug that's out first. So I think part of what we are advocating is some of these companies that are the third and fourth and fifth to market. You need to go in areas that RevMed isn't going, and so I think we're going to see more in that space. The immunotherapy part of it, the vaccine part, when you have amplification, is a little hard because NRAS is a normal protein, and so all of these proteins are normal proteins in our cells, and then when they're mutated, then you can make a vaccine against them. But if they're not mutated, if they're just high, like in your sister's case, then if you make a vaccine to that, it'll be all your cells, right? And so that's the challenge of the vaccination approach, but I think the direct targeted approach is going to be, that's on its way.
Over here.
And my question is probably going to go right in line with that because my 22-year-old son was diagnosed with MSS, and very sadly he passed away.
I'm so sorry.
He was MSS, but then he was NRAS, KRAS wild, which they told us this is great, this is wonderful 5 minutes later, they told us the cancer tricked them and got away from them, which I'm not quite sure how that happened. He had poorly differentiated cells, so I don't know how that all feeds into it. But my question again was the NRAS, KRAS wild. Is there, like, what percentage of cancers, MSS, are NRAS, KRAS wild without mutations?
So it is 60%, about 50 to 60% that are wild type for those two. And so these drugs that are going to hit broadly across wild type and normal, those drugs are going to be very interesting for us to study in those spaces too. They're going quick, we want to get these drugs into patients as quickly as possible by getting them FDA approved. And so I think that's why the companies have really laser focused on mutated patients to begin with, to just get the signal, get the drug approved. But these cohorts, meaning studying it in these other patients is coming. It's just, we need it to come as soon as possible.
Yeah, I've got 3 others that I'm constantly worried about. So I'm like, please get the science going in case one of my other ones comes with this, But thank you.
The other thing I would add to that is, as I referenced, in 21, I was looking for a trial and had looked at a KRAS targeted trial, Dr. Azad's. I didn't quite fit for other reasons. And I had this NIH trial, which was a vaccine and immunotherapy, but it was targeting CEA and MUC1. Had nothing to do with my KRAS G12V. And Dr. Marshall said to me, he's like, look, don't think that just because it's targeted, it's going to win the race. Maybe something else could work better for you. And, thank goodness he did. And that trial, now I had progression from the trial, but it was a game changer for me. And, several months later, I got to NED. And, knock on everything, have been NED since then. And so I just say that it doesn't have to be a targeted agent. We all want a targeted agent, but maybe one of these immunotherapy trials or a different thing will help you. Do we have one more question in the front here? And then that'll be our last question. Is there a microphone?
It's coming.
We're out of time, but I'm going to do one last question.
So this is kind of a theoretical question. I'm currently no evidence of disease. Been doing Signatera's from the beginning, and they've all been negative. If you have recurrences, is there ever a point where getting another tumor sample and starting fresh is a good idea?
Yeah. No, good question. And we looked at that early on in our validation studies. And what we found out is, and that's the secret sauce of how you pick those 16 variants, right? So we target clonal or truncal mutations. So they're coming from the mother cancer cells and they're propagated throughout the cancer evolution journey. The other part of it is we don't necessarily target or track driver mutations. Some of the 16 variants might be driver mutations, but that's not our goal. Most of the time, it's passenger mutations. So, for example, if someone has a KRAS mutated and they're on a KRAS inhibitor, you're going to see that that clone is going to shrink. There might be clonal evolution. A different driver mutation is driving the progression. But if you're just tracking the KRAS clone, you're going to miss it. So that's why we tried to stay away from driver mutations. And what we've seen is doesn't matter how many lines of therapy or how much clonal evolution happens in the tumor, we're still able to pick it up. Of course, no test is 100%, but we have really good concordance there.
And the other test you could do, I had my mets looked at for the tumor mutational burden because I was curious, was that growing? And would that open up better options for immunotherapy? So that's another test to look at. Join me in thanking Dr. Azad and Dr. Jurdi for sharing their wisdom.